I would like to thank for invitation to Alexander Borisovich, and I will start from the statement that my presentation will be about exposure of fetal brain to maternal antibodies and the implication of this exposure for neurodevelopment. This subject is probably a unifying theme for more than autoantibodies that I'm working on, which is lupus antibodies, and I will show you in a second an example of the effect of these antibodies in adult brain. And one of the factors which is important and wasn't discussed today, maybe touched a little bit by Professor Czeslawa, blood-brain barrier. And I will focus particularly on developing blood-brain barrier in the embryo. So antibodies that possibly are reactive to brain antigens may not participate in negative selection during B cells because B cells are continuously produced in organisms through life, and negative selections occurs in bone marrow. And unless we have unknown mechanism of brain antigen in periphery, that negative selection doesn't occur. But we know many brain-reactive antibodies, and they can possibly arise through the mechanism of molecular mimicry, or antigen spreading, or even hypermutation of immunoglobulin during protective antibody response to infection. And they can acquire, they can mediate acquired changes in cognition and behavior, as illustrated in these pictures of anti-double-stranded DNA antibodies that enter the brain after breaching of the blood-brain barrier. Anti-Dweez and anti-double-stranded DNA antibodies were created in Betty Diamond's lab, in whose department I work, by creating a double-stranded DNA nemotope that is a major antibody in lupus. I would like to point that antibody deposits after opening the blood-brain barrier with lipopolysaccharide, which is surrogate for infection, and primarily in hippocampal area, and cause neuronal damage, which results in alteration of spatial memory. If the blood-brain barrier is open with stress hormone, epinephrine, interestingly, the location of antibody deposition is amygdala, and the result is alteration of stress response, lack in fear conditioning. So what about maternal antibodies, the topic of my talk today? Maternal antibodies are delivered to developing fetus to provide protection against common pathogen, and also to help to maintain herd immunity through maternal antibodies. But in the light of developmental disorders arising in children and young adolescents, such as schizophrenia, autism, mood, and learning disorders, the example of it in the States is the recent report of Center for Control and Prevention of Disease. The current incidence is 1 in 88. A decade ago, this will be 10 times less. Genetic and environmental factors and the increased rate of survival of preterm babies obviously are contributory, but maternal antibodies are focus of attention of research recently, and they are also considered contributory. In lupus, children of lupus mothers, but not fathers, have some learning disorders as reported very early by Lahita. And here are the total numbers and controls. Mainly this was dyslexia and learning disorders and some behavioral disorders. They are confirmed by later studies. So we ask ourselves is whether these anti-DNA antibodies that cross-react with an NMDA receptor could cause neuronal damage during development if they are present in the circulation during pregnancy. This is a simple experimental design when we immunized mice and boosted immunized mice with the antigen that creates anti-double-stranded DNA, anti-NMDA receptor response. And then we mated the animals in time fashion to analyze embryos at particular time points. And the time point we selected was E15, and then the behavior of this offspring was analyzed in later time points. On the bottom you have simple serum titer. We have high serum titer, and this is a natural response in some animals that were lower titer, and this is control backbone. So I will show you a couple of the slides illustrating what happens to the brain of this developing embryo. This is a picture of E15, and what we see cortical plate thinning in the embryo that had been exposed to a peptide antibody, anti-NMDA receptor. In control, this is normal thickness of the layer, and this is an illustration. There was increased apoptotic activity. You see a tunnel assay of peptide mouse and control mouse. The other change that we noticed was increased mitotic activity, checked here by phosphon H3. That is characteristic of M phase of mitotic activity. This is present in normal development, but what we notice is the higher amount of this activity and the activity that was observed in higher level of cortical plate. This is a summary of what I just showed. This is tunnel assay, pH3, mitotic activity, and this is thinning of cortical plate. This is a ratio of cortical plate to the total width. We also did control scrambled peptides, and this is simple illustration of it. We used two peptides. This is scramble, and this is a replacement of this release motif by alanines, and both of them produce normal response. This is control mouse. This is serum titer of both antibodies. We also did tested injection of antibody that reacts with double-stranded DNA and NMDA receptor, mouse antibody R4A, and human antibody that was created in the lab. And both of them, the passive transfer of these antibodies injected at E12 and analyzed again at E15 produced similar effect as described for this peptide, and controls behave as map backbone. This is a busy slide, but I'm showing you here the tests, the physiological tests that they were performed, spatial tests, habit learning, contextual memory, tone memory, and few conditions, they are normal, and that's the point I would like to make here. What was not normal was that adult offspring had a cortical-mediated impairment in recognition task. And this is novel object when you put the mice, probably known to be obvious, where the mouse is familiarized with two identical objects, and after time delay it was put in the same chamber with the new object, and normal mice spends more time on the new object than on all. But mouse that was treated with high titer of anti-NMDA antibody did not perform well in this assay. And neither in fear condition extinction, where the tone is associated with mild electrical shock, and after time, the reaction of mice was evaluated to tone only. And normal mice learned quickly not to freeze, but the experimental mouse had a prolonged freeze. This is a summary of this essay, including the essay of geotaxic, negative geotaxic of very early pups, newborn pups, dating 18 to 16. So we had a question about developing blood-brain barrier. Do maternal antibodies penetrate the fetal brain? And if so, do they do so throughout gestation? If not, at what gestational stage are developing fetal brain capillaries no longer permeable to maternal antibodies? And can we identify endothelial cell marker or markers associated with maternal antibody permeability for the benefit of human studies? Because obviously we cannot perfuse human embryos. And what regulates maternal antibody delivery to the fetal brain? And actually, I was searching literature, and surprisingly, in spite of the importance of immunoglobulin in fetal development, there is no information of fetal antibody in fetal brain. So what we did, I labeled the antibody because, in my opinion, if one wants to test the antigen or any molecule that is delivered to the blood-brain barrier, whether adult or embryonal, one has to test this particular antibody because of specific transport system, very intricate transport system that exists on the blood-brain barrier. So the antibody was labeled with infrared dye that is very stable in vivo, important thing, and the mouse embryos were evaluated 1 hour after the injection. And what you see here is a picture of the embryos at different gestational stage. The point I would like to make here is that this resolution obviously doesn't allow us to quantify the amount of antibody that was delivered to the brain, but we see the antibody in brain area, and what we have noticed is maybe the increasing amount of separation of the antibody, which seems to be diffused early in developing brain capillaries. On the bottom of the picture is control antibody that wasn't injected, embryo that wasn't injected with antibody, and this is simply autofluorescence. We also used a cardiac perfusion using a traditional molecule that is used in evaluating of the blood-brain barrier, which is advanced blue. And on the top of this panel is embryonal head of embryo of E13, and which darkens over time. This is E15.5, and this is E17, when it's almost not visible. This is 40s slices of a brain of these embryos, and you see brain capillaries. They are red, but advanced blue spreads out at this stage of the embryo. Here at 15 contain in developing capillaries, which is more pronounced at E17. And this is similar sections of E14 embryos and E17 by comparison, again leaking of advanced blue earlier and containing at the later stage. Okay, what we did next, we perfused embryos. They were injected with these infrared antibodies. We extracted the brain and we produced brain lysate to quantify the amount of antibody delivered to the brain. And this is quantification at different time points. At E13 and E14, we have some fluctuation, but lower amount of antibody in the brain, though detectable. And here is an interesting point, because we have injected two antibodies. This antibody with the round spots is our foreign antibody that binds the brain, and the diamond one is the same antibody, the same amount, the same or similar time of injection. It doesn't bind, and I see a qualitative difference between this particular stage of development. This is after 24 hours after injection, so this is enough time to for the antibody to penetrate and eventually stay in the brain. At E16, I don't see a difference anymore, and definitely not at E17. They are the same. We have tested expression of tight junctions proteins. They are in adult brain. They are responsible for size selection, and mainly claudins. Claudin 5, I saw expression of claudin 5 from very early at E13, a very visible expression through E17. Similarly, and I'm not going to focus on also on other tight junction proteins, ZO and occludin, and the only marker that I see difference was actually the marker that was suggested to coincide with blood-brain barrier development by Buchter and his colleagues from Max Planck Institute. And this antigen disappears over time in the brain. In periphery it behaves differently. But in the brain it's very high level at E13, at E14 still, at E15 starts to disappear, barely visible, at E16 not visible in adult brain or E17. So this could be a potential marker, but this is a sort of negative marker. So it would be nice to have a marker that will indicate this ceiling of blood-brain barrier for immunoglobulin. I emphasize that in developing brain. This is glucose transporter expression in the same brain, different slicing, to show that brain capillaries at E17 and E16 are present without problem. This is a picture from the paper of Roncalli's lab when they analyzed human brains, fetal brain, and they saw a change, a quantitative change from on tight junction expression, this is occludin, this is claudin, between week 12 at week 14 and week 18. And week 18 they say it looks like mature vessels. Conclusions. In utero exposure to maternal brain-reactive antibodies can alter brain circuits responsible for cognition and behavior of the offspring. Maturing blood-brain barrier in our experiment progressively restricts maternal antibody penetration to the fetal brain. E15.5 in our hands is the turning point.